I'd like to actually start off by thanking Nick for doing such a great job introducing laser ablation ICP-MS and introducing lead isotopes as a technique to be used for Tephra chronology. So there's a lot of stuff that I don't need to go over. It's great. I actually haven't spent a lot of slides on any of the technical aspects, so hopefully everybody is up to speed after that great talk. So this is some work. I'm relatively new to Tephra chronology and Tephra stratigraphy. So this is some work that I kind of backed into by collaborating with a couple of other people at the Berkeley Geochronology Center, in particular Paul Rene and Al Dano, whose names you might recognize as argon-argon geochronologists. So this is some work that we've been doing in mainly working at facilitating some work that we're doing in Montana in some Cretaceous Paleogene terrestrial sections, and that's a photograph of this here. This is the Cretaceous Paleogene boundary here. And this is work I've been doing in conjunction, besides Paul Rene and Al Dano, with Warren Sharp and a former undergraduate student at UC Berkeley, now a graduate student at Pullman, Jessica Banasak. So before I get to the end of the slides and run out of time, I'd like to make my acknowledgments here. I acknowledge funding from the Anne and Gordon Getty Foundation, as well as some of my colleagues. Sean Mulcahy, who's been doing a lot of really heroic electron probe work, as well as Courtney Sprain, a PhD student who's led the way in the argon-argon geochronology and also the fieldwork in the Montana sections, and also Alka Tripathy and Nick Filstra for help with laser ablation and keeping the Neptune up and running, respectively. So I'll cut to the chase here. In terms of Tephra chronology, we've got obviously lots of great tools, in particular glass chemistry and crystal chemistry. These are powerful tools. They're fantastic. And I guess we're always looking for alternative or complementary ways to differentiate, to uniquely identify Tephra. You know, when field relationships fail, and we need that other method to uniquely identify these materials. Isotopic compositions of radiogenic nuclides offer some of this complementary information: neodymium, strontium, lead, osmium, hafnium, whatever you like. They're very useful. They're difficult to alter, difficult to fractionate. Often they're unique to particular sources, and often they're relatively univariant for a particular eruption. So you don't need to make dozens or hundreds of analyses. You can be pretty confident that one analysis will adequately characterize the isotopic composition of that particular material. However, the downside of that is, of course, they are expensive, time-consuming, and often consume a huge amount of sample. Now, lead isotopes might be able to get around some of those problems. For one thing, compared to other conventional radiogenic isotopes, which tend to only have one isotope to measure, or one radiogenic isotope, lead isotopes have three distinct compositional vectors. They've got four isotopes, three of them are radiogenic. They're relatively easy to measure. In part, this is coupled to the fact that there's a wide variation in isotopic composition. But the ease of measurement, lead isotopes have been measured for decades. They're relatively easy to measure, and there's a wide variation, so we can get away with a slightly lower precision than something like neodymium or hafnium or osmium. I also want to emphasize that what we're doing here is not geochronology. You can do geochronology with lead isotopes or with uranium lead or thorium lead ratios, but that's not what we're doing here. We're just basically doing pattern matching, looking at the similarity in isotopic composition from one material to another. They're also, one of the benefits here is that they're likely to have a relatively invariant isotopic composition. And I'll point out in these slides, the IC here always refers to isotopic composition. So a given source area, a given eruptive event probably has a relatively unique isotopic composition. That's great. That means we need to make fewer analyses to properly characterize an individual sample, which is fantastic when we have to pay money for the analyses. And of course, this is work that is building on the classic study, John Westgate and Nick Pierce and colleagues in Quaternary International in 2011, where this kind of got kicked off in a serious way. So a couple of things that we're really interested in. One, do lead isotopic compositions vary between closely spaced tephra at a level that can be resolved by laser ablation multi-collector ICP-MS? And two, are the distributions, if there are distributions of lead isotope compositions within individual samples, are the distributions simple enough to characterize with a small number of samples? I don't want to be measuring these, making hundreds and hundreds of measurements for each sample, because that's extraordinarily expensive, and that's not useful at all. So I'm going to first start off, I realize that there's a broad range of interests here, so I'm going to start off by actually introducing lead isotope geochemistry so that everybody is up to speed. So lead is composed of four stable isotopes: lead 204, 206, 207, 208. 204 is a primordial lead isotope. There's no radiogenic additions to it. The Earth has the same budget of lead 204 as it did when it accreted. whereas lead 206, 207, and 208 are continuously being added to by radiogenic addition from uranium 238, 235, and thorium 232. That looks kind of like this. On a whole Earth scale, we started off with this amount of lead and these relative amounts of uranium, and we've added 206, 207, 208, and subtracted 238, 235, and 232. How that looks in terms of a common way of plotting lead isotope compositions, this is a pretty common way to plot lead. 204 is the denominator on the x-axis and also on the y-axis, and that's the invariant lead isotope, the one that's not created by radiogenic addition. 206 is created by radiogenic addition by decay of uranium 238. 207 is created by radiogenic addition through uranium 235. So if we had, say, on a whole Earth scale, if the Earth accreted with an initially homogeneous lead isotope composition, and then it differentiates into, say, 3 different reservoirs like the crust, the upper crust, the lower crust, and the mantle, with different chemical compositions, different ratios of uranium to lead. They'll evolve along these, I think you can see this, curved trajectories as a function of time. So in 4.5 billion years, you can get relatively large variations in the isotopic composition of lead isotopes. And that's what the overall topology of lead isotope compositions look like. We also have lead 208, which I don't plot on here, but in general works in a very similar way. So that leads to relatively large variations in lead isotope compositions. So long-lived differences in the time-integrated chemical compositions lead to different lead isotope compositions for different geological provinces. This is just a compilation of igneous rocks, lead isotope compositions of igneous rocks from around North America. This is a huge variation. This tiny little box here is 0.1% in either direction, and that's the type of that's an easily resolved difference in isotopic compositions using modern techniques. So you can see all of this variability is very, very easily resolved. Now, going to a smaller scale, okay, so we know that we've got a large variability in lead isotopic compositions. Looking at a smaller scale, on the scale of a single eruption, what do we expect to see? Now, this is some data from a really, really excellent paper by Wolf and Ramos, a geology paper in 2003. This is 206, 204 versus 208, 204. Very similar to the last plot. And here, this blue cross is the same size as that tiny blue square here, okay? We're looking at a totally different scale. And these are individual phenocrysts of sanidine from the Bandelier Tuff. So this is all from one single eruption. And so they do see some variability, but it's very small and unlikely to be picked up by the laser ablation MC-ICP-MS techniques, by a technique that has a lower precision. The technique that they used was a very expensive, time-consuming, but ultra-precise and accurate technique, a solution-based measurement. So this is good news. This means that the variability within an individual unit is probably going to be very small. Okay, so all right, so we've established that it's, there's probably a good reason to have a crack at this for Tephra chronology. And here's the technique that we're going to use. This is laser ablation multi-collector ICP-MS. I put the MC in parentheses because it's not absolutely necessary to use a multi-collector instrument. As Nick and colleagues have shown, they can get really good, very interesting, scientifically useful data with single collector instruments. And I'm not going to go into a lot of detail on this. We use a laser to sample solid materials, and that's transferred to a mass spectrometer where it's ionized and analyzed. So the benefits of laser ablation ICP-MS over conventional bulk techniques are that it's very fast. You can do a single analysis in 90 seconds, 120 seconds if you like. It requires very, very little sample preparation. You just need to crudely mount the materials in a standard 1-inch epoxy puck, do a crude polish, and Bob's your uncle. It doesn't require special clean labs or enriched or synthetic isotopes, which is great. These things are expensive, they're time-consuming. And with the techniques that we employ at the BGC, the precision approach is 0.1% for lead isotope ratios that include 204, and 0.05%, that's 500 ppm at 2 sigma, for ratios that don't include 204, so the 208, 206, and 207, 206 ratios. And I'll point out actually here that relative to the types of analytical conditions that are used to analyze trace elements in glass, we're actually using much larger pits on the order of 90 to 100 micron diameter. And accuracy may be commensurate with precision when we analyze secondary reference materials. I say maybe because The secondary reference materials that I have, we actually produce data that's more precise than the TIMS data that we use as the nominal reference values. So it's actually, at this point, it's very difficult to tell how accurate we are because we actually do a little bit better precision-wise. Not necessarily accuracy, but certainly precision-wise. The downsides of this technique is that it requires complicated and expensive equipment. It requires a good laser and a multi-collector ICP-MS. These are not trivial pieces of instrumentation. Well-characterized reference materials are necessary. For the most part, we've got that covered with the NIST 61X glasses, 610 and 612. Matrix matching is, of course, a potential problem. I haven't seen that crop up in the work that we're doing, but it's potential at high precision to be a problem. And, of course, it requires samples with enough lead to measure. So I haven't specifically talked about the materials that we're using, but for the work that I'm showing here, we've exclusively focused on feldspars, mainly sanidine, because these provide a direct link to geochronology. Okay, I'm working mainly with argon geochronologists, so they're very concerned about the sanidines, about the feldspars. And so we already have large quantities of sanidine separates. They're also less easily altered than glass. The other important thing is that the main regions that I've been working, we actually don't have glass either because the sections are too old, like in the case of the Cretaceous-Paleogene boundary, where the glass is all shot, or in some places where Aldano is working in East Africa and the Tephra have erupted into alkaline lakes and the glass is again, very, very altered. For more ancient materials, it's actually very useful that the feldspars don't have a high lead-uranium ratio. So you don't have ingrowth of uranium after this has been deposited. That's something that you'd need to correct for. So that's also something to think about when you're working on something that's, say, more ancient than the Pleistocene. This is a typical analytical session, something like 7 hours, where we're analyzing samples here interspersed with secondary reference materials and then calibration reference materials. And I'll just mention to Steve that, yes, I do plan on publishing my analyses for the secondary reference materials. So I just had a note here on data visualization, which I think is an important part of everything that we do. So for one thing, because we're working with compositional data, there are 4 isotopes. We should actually be plotting everything on a tetrahedral simplex, but that's a bit more complicated. So we're going to pretend here that we're working in the domain of real numbers, just like everybody does in the electron microprobe world. But because there are only 3 independent compositional vectors, all information on lead isotope compositions can be contained in 2 scatter plots. Here we go, 207, 204 versus 206, 204, and then 208, 204 versus 206, 204. These 2 plots contain all the information that's necessary to define a unique lead isotope composition. So another thing that, in terms of data visualization, is that we have these error ellipses, and we produce a lot of data. And multiple error ellipses, a profusion of overlapping error ellipses is very cumbersome to plot. It's very awkward on the eye, and it makes it very hard to read data ultimately. These ones not so bad, but and others it is. So what we've been doing, I hope these lines show up for you, but what we've been doing at the BGC is using kernel density estimation. So what we do is we treat each of these little analyses. Each one of these has its own error ellipse, and it's a little bivariate Gaussian distribution. So what we do is we sum all of these bivariate Gaussian distributions, and they get this, in this case it looks like a Gaussian distribution on its own, but when the distribution is non-Gaussian, you get this sort of lumpy, bumpy kind of thing that's a useful estimate of the population of analyses while respecting the analytical uncertainties. You kind of see this lumpy bumpiness here. And so what we do, instead of plotting all the contours, we just plot the 95% confidence interval. And that gives us a useful way to plot a lot of data on a single plot. And that's kind of what we've got here. For this NAVDAT data, this light blue kind of potato-looking thing is actually the 95% confidence interval on the kernel density estimate for the NAVDAT data. So it's a useful way to identify the data without having to show all of the error ellipses. Okay, so now we'll get to some data now that we're set. We've got our analytical technique. We know how we're going to display our data. We all are experts on how lead isotopes work. We're going to go to the Cretaceous-Paleogene boundary in the Hell Creek region in Montana. This is located in Montana, just south of the Fort Peck Lake Reservoir. It's kind of in the middle of nowhere in Montana. But it's a great place for vertebrate paleontology. In fact, the Hell Creek region is one of the best places, the best terrestrial records of environmental, floral, and faunal changes across Cretaceous-Paleogene transition. Unfortunately, because it's terrestrial, there are rapid lateral facies variations, and it's very difficult to know where you are within the stratigraphy, right? And that's very important when you're looking at, say, tens or hundreds of thousands of years of evolution of mammalian fauna after the Cretaceous-Paleogene boundary, after the mass extinction. So we also, like at Mount St. Helens, we have this alphabet soup-based lithostratigraphy. Although in this case, they've put some effort into making it alphabetical, which is kind of useful. Although in many cases, there are multiple different horizons that have been nominally named the same. So there are multiple Z-cols, for example, which do at times have different ages or have indistinguishable radiometric ages, but are known stratigraphically to have different relative ages. And so these are some of the problems that we're trying to sort out. These coal seams that are used for correlation have Tephra in them. I think I've got a picture here. Yeah. So this is a picture of one of the coal seams. This is about like just slightly less than a meter thick. And you can see up here. This is the same picture right here. My colleague is pointing at one of these Tephra that we use to date and to correlate the coal seams and the stratigraphy in the Hell Creek region. So this is the kind of thing that we're dealing with, these sub-centimeter thick tufts inside of the coals. So the coals are used for stratigraphy, and we use the tufts to date them and to sort of uniquely identify them. So I'm going to just scroll through some of the highlights and the lowlights. The good, these are the Z-coal associated tufts. These are the ones right around the Cretaceous-Paleogene transition, super important. So the idea is whether we can distinguish tufts in the various distinct Z-coals so that we can uniquely identify them for the paleontologists. So this one worked out really well. This is one of these tufts associated with a Z-coal that's also associated with an iridium anomaly. So these ones are right at the Cretaceous-Paleogene transition. These are four different samples from four different localities. And there's really, really good overlap between 3 out of the 4, and then one of them, this green one here, MK13.3, has a lot more scatter. We assume that these are xenocrysts, possibly contaminants either induced during the sampling process. These are very difficult to sample cleanly, or during the eruption and deposition process. It's not entirely clear where these come from. Looking at a set of tufts, one tuft from another Z-coal, we've got a lot more samples, and there's great overlap between them. I really like the way that these things overlap in lead isotope space. Again, with some of the samples, there's a few outliers. Again, we assume these are xenocrysts, some sort of contaminant, either natural or induced during the sampling process. And if we plot all these, so we plot those two along with 2 other tufts from these 3 units, 4 diagnostic tufts from 3 horizons can be very, very easily distinguished. So this blue one and this red one actually have very slightly different ages. So even though they're close in isotopic composition, we know for sure that they're actually different tufts. And then these ones, this black unit here and this green unit here are actually a doublet that are found in a single coal. So these are actually just a few centimeters apart, but they have a widely dispersed lead isotopic composition. For those of you with keen eyes can see that there's some dark spots here right next to this red blotch, this dark spot that should be related to these. And I'm going to call this a problematic sample. This is one that we thought was associated with this package of tufts, but has a clearly distinct lead isotopic composition. We actually went back to the field, and one of the things that we did in the field was try to sort this problem out, because it's actually very important for the geochronology of one particular section. So we've used this now to inform our sampling strategy and our geochronology, and perhaps the way that the samples were mixed up in the lab. Okay, so those are the good ones. So this is good. Everything, it requires very few analyses from a bunch of different tufts to uniquely identify these tufts and help out our correlations. So this is now the bad. So that was the good. This is the bad, and this is a sample that we knew coming in might be problematic. These are argon-argon total fusion ages. This is a number of analyses by Courtney Sprain. These are from a publication that's now in press in GSA Bulletin. So there was a juvenile fraction at about 65 million years, and then there's a fraction of xenocrysts, of inherited sanidine grains. And so when we measured them by ICP-MS, we see the same thing. We see a lot of scatter. I only measured 4 analyses, but there's a huge amount of scatter, obviously, in these. And we would be able to define a juvenile population, or at least a dominant. We would be able to capture this population if we made dozens or perhaps more than dozens of analyses, but that's not something that we were really all that invested in. So this one is a bit of a wash, I think. Although it's perhaps unique because it has a lot of xenocrysts, it's not clear whether that's a function of simply of that particular sampling locality. And so I went with the good, the bad, and it was supposed to be ugly, but this is actually very interesting, so I just crossed that out. So this one is another one that we knew going into it that was going to be complicated. So from the argon data, these are some compositional results from the argon-argon data, where there's clearly a bimodal bulk compositional distribution. That's the calcium-potassium ratio. Whereas this is hard, this is basically impossible to read. But this is the argon-argon data. It's unimodal. So it's clear that these phenocrysts are all actually juvenile. So this is some compositional data, and this is some electron probe data. There appears to be a trimodal bulk composition, mainly in the potassium-calcium ratio. So we know that these things are not uniform in their bulk composition, but they are uniform in their age, in their ages. So we infer that these are all juvenile. These aren't contaminants. And actually, the lead isotope composition is actually very interesting. It was kind of neat to do. So the lead isotope compositions. So the plot up here and the plot down here are the same axes, but the one below is just in this stippled area here. There are basically two populations, one with low lead concentrations, that's why the uncertainties are so large, but a more radiogenic composition. And then there's a second population that form a more or less continuous trend from a 6.4 of 18 to about 18.6. Okay? And then when we relate those directly to chemical data, so these are analyses that have coupled EPMA and lead isotope compositions, the low albite samples tend to be the ones that have these low lead concentrations and highly radiogenic 6.4s. And the rest of the samples appear, I don't know whether this is convincing to me, so maybe it's not convincing to you, but there's some sort of crude correlation with calcium content with the 6.4 ratio. Again, I'm not totally convinced that it's there. But this is all juvenile. I think this is all primary, associated with magma chamber processes. So something like assimilation or magma mixing within the magma chamber, which as a, with my other hat as a petrologist, I think is very interesting. This is actually a very large range in isotopic composition. And that's kind of unique to magmatic systems, I think. So to summarize and to plot up a whole bunch of data, we're able to define unique lead isotope compositions from many individual tephra in the Hill Creek region of Montana, and this will help us aid the paleontologists in their search for stratigraphy. We can also do something really interesting with lead isotopes, like what was shown before. We've tentatively defined a unique source in Idaho in the Bitterroot batholith based on the lead isotope composition, so several hundred kilometers away. This is the Bitterroot batholith in blue and some of the Montana tephra data in red. They don't exactly overlap, but the Bitterroot batholith is incompletely sampled, so, and it's a better match than anything else in the region. So, and it's roughly the same age, which is consistent with it being the source area. So it's very pleasing. Okay, so where do we go? What's the deal? The technique is fast and straightforward. I'd say if you have a very simple population, it takes about 30 minutes to reasonably characterize it, a single sample. And so if you have a 24-hour session, you could probably characterize 30, 35 samples in a day. It's an expensive technique relative to EPMA, but if you leverage the amount of material that you can go through, then it's not so bad. It appears that many eruptions have distinct lead isotope compositions, but we don't have a lot of data from serial sections through single eruptive centers. Some or many eruptive products have variability in lead isotope compositions that we just can't resolve, which is great. They're essentially invariant for all intents and purposes. I think it's a great complementary technique. It's better for K-feldspar than for glass, particularly when you have large samples. I'm dealing with somewhat larger spots, although I think we could probably get down to the 80, 70, maybe 50 micron range, particularly if you have a multi-collector with multiple ion counters. All these, for those technically inclined, all of these measurements were made with multiple Faraday cups with one ion counter measuring mercury. Glass is certainly possible. And the upside is that phenocrysts and glasses should have the same isotopic composition. It's not something that's discriminated between phases. So you should be able to mix and match phases. So you could measure the lead isotopic composition in feldspars and then in the glass and see, and they should match if they're from the same eruption. Although that's something that needs to be more validated. So what do we need going forward? Better constraints on total lead isotope composition of single volcanic setters. That'll improve our source tracing and our knowledge of how things evolve over time. So serial sections through individual volcanic sections. Improved secondary reference material characterization. It's not It's not a good thing when your primary technique is less precise than the microbeam technique that you're using. That's sort of suboptimal. And of course, consistent data treatment and reporting strategies. If anybody's really interested in the way that we did the data treatment, I've got lots and lots of slides on data treatment. So if anybody's interested in that, I can talk to them afterwards, particularly how the uncertainties are estimated and treated. And I think I'll just leave you there. Thanks.